All right. So today we are going to continue talking about vaccines. Because it's the end of the semester and everything awesome happens at the end of the semester, I forgot to hit record on Friday. So I do still need to re-record Friday's class, but we will persevere and move forward. We went through the first section of this on Friday, and we left off talking about vaccine types. We specifically talked about two different vaccine types on Friday, the live attenuated vaccine and the inactivated vaccine. And we saw both the pros and the cons of those vaccine types. Those vaccines are sort of the classic vaccines. If you ask someone who does not have a much immunology background to explain to you details of how certain vaccines work, they're probably going to explain to you or imagine in their heads live attenuated and inactivated. I certainly could have said that before 2020. There's one other one that's kind of in people's heads now, but these are the ones that everybody had been thinking about. There are a few other types of vaccines out there. And so the sort of two old classics are the ones that I told you about. But in many of the other types of vaccines, we take just a part of the microbe and use it for the vaccine. So with the live attenuated, we had the whole microbe, but it had been weakened. With an inactivated, we had the whole microbe that had been killed. With all of the other types that we're going to talk about today, we're going to take a piece of the microbe and use it in the vaccine. Sometimes we could get that piece by making a whole bunch of the microbe and then breaking it up and purifying out the piece. Other times, we make the piece in the laboratory using molecular biology and sort of protein and other kinds of sort of chemical techniques. Depending on the particular type of molecule or the kind of piece of the microbe that we are using, we could have either a toxoid, a subunit, or a conjugate vaccine. With a toxoid-based vaccine, instead of giving you the entire microbe, we give you a modified version of a toxin made by that microbe. The idea here is that the toxin is the thing that actually causes disease. The rest of the microbe doesn't do anything to you. And so instead of having you make an immune response to the whole microbe, we just make an immune response to that dangerous toxin. Oftentimes, we try to make an antibody response so that we have antibodies to neutralize the toxin. And of course, you can see that that toxin had to be modified in some way. If I just injected you full of bacterial toxin, you would not be very happy with me. One thing that we can do is use a conjugate vaccine. A conjugate vaccine actually works very much like something else we've already talked about in this class already. And again, for reasons why I don't understand, this next image that I'm going to show you of a conjugate tries to combine a conjugate and a toxoid. And yes, the vaccine that they're using as an example does have that combination, but I don't want you to, they're not always combined. They don't have to be combined. I wish they didn't combine them on the slide. With a conjugate, again, we are going to give you a piece of the microbe. And in the case of a conjugate, the piece of the microbe is a polysaccharide, a carbohydrate. As we talked about, see, pretend this doesn't say toxoid right there. As we talked about earlier in the semester, we can make antibodies against polysaccharides, but there's this problem with T-cell recognition. And so if we actually want to have a vaccine that contains a polysaccharide, we need to link it to some kind of protein, a protein carrier, in order to give us some T-cell epitopes so that we can get T-cell help. This is that linked recognition process we talked about before. Here they're saying, what if you got a 2-for-1 and you took a polysaccharide from some bacteria and you took a toxoid from another one? That was your protein, and then you actually got 2 vaccines in one. Yeah, that's what they do in this particular case. It doesn't have to be. It just has to be some kind of protein linked to that conjugate. You can also see that your diphtheria vaccine and your tetanus vaccine that you have received are both toxoids. If you've received a streptococcus or an H. influenzae, those are conjugate vaccines. As I would argue is pertussis. The other really big type of vaccine that is used is a subunit vaccine. And the way that we work with a subunit vaccine is that we use some kind of protein antigen from the microbe. So to give you an example, with my vaccine types back here, here you can see the entire SARS-CoV-2 virus. A subunit vaccine of SARS-CoV-2 might just be a protein, just the protein including spike. So we might make the spike protein and give you the spike protein. Sometimes we'll even give you a piece of the spike protein. But here and here and here, you can see different, just making the spike protein that we could use. And one could do that by taking the virus and breaking it apart and purifying out all the spike proteins. That seems kind of easy. Also is a little bit of a problem in that A, if you didn't actually break down the virus enough, you could still infect people, right? Like you could still have virus parts if you didn't do a really good job of purification. It also means in the case of some dangerous virus, you would have to grow a whole lot of dangerous virus in the lab before you broke it up. And so that part could be dangerous in and of itself. So generally what we actually do now with all of our subunit vaccines is we use molecular biology techniques to clone the DNA, encoding that protein of interest into some kind of cell, perhaps a bacterial cell. Sometimes it's done in some other cell types. Sometimes it's done in mammalian cell culture, like we're using in the lab. There are a few different kinds of cells you can use. But you basically just put a plasmid with the gene of interest into the cells and have those cells make the protein. And then you have large amounts of that purified protein. And you never even had to have the real virus in your lab. So there's no possible way these purified proteins are going to infect someone or cause disease. They're not going to get out of control and start replicating because they're just purified proteins you made of one part of the virus. There are 2 famous subunit vaccines that you may have received. The hepatitis B vaccine was the first major subunit vaccine that was described and made in this way. It is made in proteins coming from insect cells. The other one is the HPV vaccine, which is also a purified protein. If you have heard of the Novavax SARS-CoV-2 vaccine, the Novavax SARS-CoV-2 vaccine is a subunit vaccine. I mentioned to you last time there are no SARS-CoV-2 live attenuated vaccines on the market. The Sinovac SARS-CoV-2 vaccine that was developed in China is an inactivated. Novavax is a subunit. But inactivated vaccines, subunit vaccines, really toxoids, conjugates, all have one big problem in common. I think about this problem most acutely when I think about subunit vaccines, but we can see it with all of these different vaccines. When we give someone a live attenuated vaccine, which is shown sort of on the bottom left here, that live virus is able to reproduce or replicate. And so the amount of virus we put into the person increases over time. And it increases over time until it drives an immune response of a really great size. With an inactivated or killed vaccine, or a purified protein, or any other purified component, the amount you put in is the amount you get. It cannot reproduce in the body to amplify the dose. And so you may very well have to give more doses or booster doses in order to amplify that response and get that response high enough to lead to protection. With the inactivated and subunit vaccines, again, we don't see infection. They're not going to, get their way into cells very easily. That means they may not be presented on all of the MHC types very easily, particularly on MHC class 1, which, if you remember, gives you or presents cytosolic peptides. So you might not make the best ever CD8 T cell response with these. If you only need an antibody response, who cares? But if your goal is a CD8 T cell response, these types of vaccines may not be ideal. And further, and again, this is true of all of these, but it's the biggest problem with a subunit. If I make a whole bunch of purified SARS-CoV-2 spike in the lab and inject that SARS-CoV-2 spike into you, there is nothing to stimulate the innate immune response. Purified protein. There's no PRR stimulate. There's no MAMP. It's just a whole bunch of purified protein. Basically, you're giving something to the innate immune response, and you gave nothing to the, you're giving something to the adaptive, and you gave nothing to the innate to help turn on a good adaptive response. Whereas with the live microbe, you've got innate responses and adaptive responses because you've got plenty of PAMPs. And so often, we have to add some additional ingredients into our these vaccines. This additional ingredient is called an adjuvant. And so an adjuvant is something that is added to improve the response to the vaccine. There are different adjuvants that different vaccines use and that different vaccine developers think about. The basic idea of the adjuvant is something that you add along with the antigen to give you a more robust immune response with less antigen. One way that those adjuvants can work is that they can have physical effects on the antigen in terms of either its stability or its localization. And so one thing you can sort of think about is that there's one major group of adjuvants that basically are like a whole bunch of oil that go around the antigen so it can't get degraded as easily. So proteases and nucleases and everything can't chew it up as easily. So it makes it last a little longer in your body. Or makes it go to a particular location in your body. There are some adjuvants that can influence antigen presentation, but there are many adjuvants that specifically work to induce innate immune responses or provide PRR signaling. The field of trying to understand adjuvants and what adjuvants to add to vaccines is a very huge and hot area in trying to understand vaccines, because if we can give you a lower dose or fewer shots and have it work better, that'd be awesome. But adjuvants are really tricky. As you can see, one of the big perks of adjuvants is that they can induce an innate immune response and PRR signaling. So let's imagine that I found an amazing adjuvant that can induce really good innate immune responses and PRR signaling, and I inject it into you. How are you going to feel? You're going to feel, yay, my vaccine worked, but besides that, you're going to feel awful. You're going to have a fever. You're going to have aches. You're going to have a lot of side effects. As you increase this innate immune response and PRR signaling, you're going to start to feel more side effects. So good news, it's going to help you make a better adaptive immune response. But bad news, it also is going to give you more side effects. The US FDA is actually pretty strict about adjuvants. We have relatively few adjuvants approved in the US. There are more adjuvants approved in other countries. So for example, the flu vaccine in Europe has an adjuvant in it that we can't use in the US. And a lot of it has to do with kind of what is the level of innate immune response / side effect that they're willing to approve. So this is a huge area of research. So it used to be, when I started teaching this class, those were the types of vaccines that existed. At least those were the types of vaccines that existed and that you had received. Of course, in the past couple of years, that has changed a little bit. And so I need to, of course, talk about that as well. And so what you can think about here first is this idea of the subunit vaccine for, say, SARS-CoV-2. So what we could imagine is we could say, I know that in coronaviruses, the most important antigen is the spike protein. I know immune responses to coronaviruses are all about spike protein. And people have known this for quite a while. And so instead of maybe using the entire virus, because I might feel that's risky to do a live attenuated, I'm going to make a purified version of the spike protein. And that would be a subunit, just like we talked about. Because that's really the antigen that matters. And my problem is, well, I don't have any PRR stimulation. I probably have to use an adjuvant. It doesn't mean inside the cytoplasm of a cell, so I'm not going to get good CD8 T cells, but it's pretty safe. So the idea came with, instead of using the whole protein, let's use nucleic acid that encodes this protein. One could use DNA, one could use RNA. I will fully admit this is a super weird slide for me. In graduate school and for much of my career, I spent a whole bunch of time working on DNA vaccines, and if you had asked me to bet in 2019 what kind of nucleic acid vaccine would be the one, I would have told you DNA. Clearly I was wrong. Because in fact it turned out that the mRNA vaccines have worked out much better and are actually now in the clinic. And so basically what happens is that this mRNA is generated. It's the mRNA that's encoding the spike protein. And that mRNA is put inside of a lipid particle. You may have heard about the nanoparticle. It's just a lipid that is circling the mRNA. What that lipid does is it helps merge with the cell membrane to put the mRNA into cells. It's going to put the mRNA into a few muscle cells in this area, as well as into dendritic cells that happen to be in the skin. And when that mRNA gets in the cytoplasm of the cell, just like other mRNAs that are already in your cell, that mRNA will get translated, that you'll have some protein produced. You might get some protein on the surface of the cell, but you'll also get some versions of that protein degraded and presented on MHC class 1 and MHC class 2. And so the muscle cell, as well as the dendritic cell that has picked this up, will be able to present on MHC class 1 and 2, as well as making antigen to stimulate antibodies. Because this is nucleic acid, it can itself bind to PRRs, so we don't have to worry about an adjuvant. The lipid does actually adjuvant as well, but the nucleic acid can adjuvant by itself. There are a bunch of different changes made to the nucleic acid. It's actually not just standard mRNA. There are a bunch of chemical changes made to influence stability and things like that. These scientists who are shown at the bottom, Drew Weissman and Katlin Carrico, really kind of worked out some of the ways to generate exactly the details that are needed for that mRNA. One thing people often ask, or sort of people that feel a lot really uncertain about with regard to mRNA vaccines, is a little bit about sort of some details of the history of mRNA vaccines. So let me just tell you a little bit. mRNA was first described in 1961. Big deal. And there were lots of discoveries about mRNA biology. The idea that you could put mRNA into a cell and get an immune response was actually made in 1993. So you guys still aren't even born yet. And people started talking about doing mRNA vaccines in the 90s. And so you can see from 2002 to 2020, lots of different human trials of different types of mRNA-related vaccines. So sometimes when people are like, oh my gosh, this happened in 5 minutes. No, it didn't actually. People have been working on some of these things for a long periods of time. These are other mRNA vaccines that have gone into clinical trials. Note that some of them, these are the publication dates. And you can see there's publications in 2008, 2009, of all of these things, each of which you can see are for cancers. And so mRNA vaccines have particularly been under development for cancers, as cancer vaccines for a very long time. And so we do know a lot about the safety profile of these vaccines from these mRNA vaccines in these clinical trials. So you can notice that. You can also notice these are some of the other mRNA vaccines that have been generated against different infectious diseases. Again, you can see the year that those vaccine trials were started, as well as the number of individuals who were enrolled. And so you can see that there are mRNA vaccines against rabies, was done in clinical trial in 101 people in 2013. So again, there is actually quite a bit of a history of the use of these vaccines. So the question that will often come up here is, okay, so if there has been a rabies vaccine in trials with mRNA vaccines since 2013, why is it that the first mRNA vaccine that we ever received is the SARS-CoV-2 vaccine? What took so long? Why haven't there been all of these other mRNA vaccines around? And the real answer to that is money. Once you do your rabies vaccine trial, you finish your phase 1 rabies vaccine trial, you then have to say, I want to invest in a rabies phase 2 vaccine trial. Will some investor give me the money? How much do you think in 2013 anybody cared about making a rabies vaccine to invest in your phase 2 trial? Do you think it was going to be easy to get the money? Was there like desperate big world need to make sure we make it right now? No. And so most of these, as well as some of the ones on the previous slide, though again we'll talk about cancer more later, had decent results, but didn't have money kind of pushing them fast through the pipeline. You know, this pipeline usually takes 10 plus years, and part of it's because there's a lot of money raising steps that have to happen. And all of these got bogged down in the money raising steps. Fortunately, the SARS-CoV-2 vaccine did not get bogged down in the money raising steps. This story is so fabulous. So this is Kizzmekia Corbett and Barney Graham. They are the scientists behind the Moderna vaccine. Caitlin Carrico and Drew Weissman are part of the Pfizer team. These guys are really the ones who were at the NIH and who really did the Moderna vaccine. They had been working on mRNA vaccines and vaccine development for a long time. They had been working, they're like friends with Fauci before, for a long time. They have been working on the MERS vaccine for a long time. And they decided, they raised a whole bunch of money to do a MERS vaccine trial, right? To sort of see if they could get this very specific MERS coronavirus vaccine to work. They raised all the money. They were sort of really going to start when they got back from Christmas break, the end of 2019, beginning of 2020. They had the whole thing built out. And then, I believe it was January 8th, they get a phone call from Tony Fauci. Hey, you know that trial you're starting? Could we maybe sub out MERS and put in the SARS-CoV-2? I mean, everybody knows the spike is the most important thing. You were going to make a MERS spike vaccine. Could you sub it out? Within 24 hours after the sequence was online, they had it in their platform, and they basically took the pre-existing trial that they were getting ready to do and shifted it over. There's one book called Breathless by David Quammen. There's another one, A Shot to Save the World, by an author whose name might be Zimmerman, definitely starts with a Z, that actually goes through all of these steps, as well as the steps that Caitlin and Drew were doing and all of that kind of stuff. So part of this was just, they were designing this vaccine because they were trying to be prepared against other types of microbes. This great paper. And part of the reason why this particular vaccine was made so quickly compared to other vaccines has to do with an engineering principle. So if you ask engineers, they will tell you that whenever you want to make a product, you can pick two of the following three things in making your product. You can make that product fast, you can make that product well, or you can make that product cheap. And you get to pick two of the three. And so when we made this vaccine, we picked fast and we picked well, and we did not pick cheap. Basically what happened here is that everybody put in all the money early. So all those steps I told you about in terms of, well, you do this trial and then you have to make money and ask if you're allowed to do the next step. Yeah, no, they just put forward the money. The US government put forward the money at the beginning and said, we're going to go for this. If the vaccine fails, we're going to have lost a ton of money, but we're going to basically start using the information we have, like the information Kiezmikia had from MERS-CoV-2, to basically sort of skip some of the original stuff, and then do all of these phases overlapping, and by paying huge amounts of money to do them fast. Because unlike that 2013 rabies vaccine, there was a world need for this, and we needed it right now. And so that really kind of moved this process forward compared to the traditional process. Just to give you some idea of how these data look. So these are actually some of the data from the Pfizer vaccine trial. In this paper, they used 2 different doses, and they used a whole bunch of different things. So they've got a lot of, so I'm only showing you some of the data just to make your life easier. Here are the people who got placebo, and here's how much antibody against spike they made. These are people in the 18 to 55 age group and how much antibody they made with 3 different doses. The middle dose is actually the dose that was used in the end. And so you can see how much more antibody people made. Here you can see elderly individuals. Same thing with the 3 different doses. And here in black, you can see human convalescent serum. So those are what antibody levels were seen in people who actually had COVID. One of the things to notice about these data is how wide this spread is for those individuals who had COVID compared to the people who got vaccine. So yeah, some people who get COVID make a lot of antibodies, but some don't. Everybody who gets the vaccine makes a lot of antibodies. So if you were in this group, you're sort of playing dice or something about if you're going to have a lot or a little. If you got the vaccine, you're definitely going to have a lot, is what they showed in these data, leading to the efficacy as is shown here. And we talked about the efficacy data last time. Just to give you 2 other pieces of information. These are, again, the antibody levels in infected individuals at different time after infection or after vaccination. So again, you can see that the people who got antibodies from the vaccine had way more antibodies than those who had infection in this study. This is looking specifically at antibodies against spike. If you look at antibodies against N, which is an antigen that's not in the vaccine, it's only in the actual virus. People who got the virus made antibodies against it, and people who got the vaccine did not, because you only make antibodies against the antigens you saw. So this antigen N is in the virus, but not in the vaccine. Because remember, when we make the vaccine, we're just using a piece of the virus. So much I could say. I could talk about this slide and this study for like a year. But yes, Carney, you had a question. That is a great question. I think that people are still sort of figuring out some of the details of what's up with the DNA vaccines, why they haven't come as far as the mRNA vaccines. I think some of it has to do with the modifications that are made in the mRNA, but I will be perfectly honest with you. That is a question that I myself have spent a fair amount of time in the literature thinking about. So I'm not sure that I can give you an answer, and yet I spend too much time thinking about the data on that. Let's see how to describe this. This slide shows something called antibody-dependent enhancement. So what has been seen is that in some cases of virus infection, I will admit I have 2 slides about this study, and I'm kind of wishing I had the other one here, but that's okay. So we have antibody-dependent enhancement. It's a possible negative thing that can happen with antibodies. It was argued about in some places on TV, and it was one of those times where I would see the news and I'd be like, excuse me, what are you saying about immunology? Like on this news station, I never expected in my life that I would be hearing such things. Basically what can happen is the antibodies can opsonize virus particles and help the virus into cells and help the infect. And what this figure shows is that antibodies from COVID patients can in fact enhance, while antibodies from people who were vaccinated against COVID cannot enhance. So the point that is being made here is that the antibodies are qualitatively different. The antibodies that are coming from the vaccine are in fact better than the antibodies that are coming from infection, because the antibodies that are coming from infection have this potential bad side effect. The antibodies from the vaccine are not. So basically what was shown was that the difference in the antibodies is in the types of sugars that are added to them, the type of glycosylation that is added to them. When the vaccine induces antibodies that have this type of molecule, there's this little red triangle. It's a fucose. And in infection, perhaps because of differences in germinal centers, differences in helper T cell subsets, all that business, there's no red triangle. There's no fucose. It's afucosylated. That changes binding to Fc receptors, and the antibodies that were made during infection can actually lead to this problematic inflammatory response, while the antibodies that are made during vaccination cannot. And sadly, I don't have the exact figure I would like to show you in an easy way, but these 2 papers describe this really nicely. And I will have to make sure I remember to switch that slide in the future. Okay. There's one other really key thing that I want to show you from some of the vaccine data. Some people have asked me, during a talk I gave earlier this semester, someone asked me what was one of the most interesting pieces of immunology I'd heard in the past year. And realistically, this is pretty much near the top. You're looking at neutralizing titers. So basically, how much neutralizing antibody is there in these different patients? And here you can see a person who was infected at 1 month after infection. They have some neutralizing antibodies, specifically against the normal strain of COVID. This is the original strain of COVID, and there's some neutralizing antibodies. If we look at people who got 1 month after a vaccine, they've got some neutralizing antibodies. In fact, they're a bit higher. If we look 6 months later, or 5 months later, you can see that they go down. Not a shocker, right? Yay for contraction. And then we look at what happens after boost. And so this person got infected and then got vaccinated. This person got vaccinated and then boosted. And you can see we get an increase in antibody levels in both cases. Hooray! But the other data are where things get really interesting. In the gray, we're looking at the original SARS-CoV-2 virus, the very first one. In red, we're looking at a version of SARS-CoV-2 that was generated in a lab that was ideally to sort of test evading or avoiding antibodies. Oh my gosh. After your infection, you don't make antibodies to this different one. The antibodies are specific to the one you got infected with. Similar, when you got your vaccine, you got a vaccine against the original. So you don't make good antibodies. Blue is Omicron. Oh my gosh. You get infected, you don't have good antibodies against Omicron. You get vaccinated, you don't get great antibodies against Omicron. However, when you see that antigen again, either in a vaccine after recovery or in a boost, suddenly your immune response increases against these similar viruses. So you get what we seem to see is this broadening of the immune response over time. So now not only can you recognize the original virus that you were infected with, you can also recognize some of these other variants. And we've seen this broadening in many, many studies. It seems as though this is related to the germinal center reaction ongoing and allowing for additional hypermutation to allow that response to broaden. But I don't really think we understood this level of broadening until we started seeing some of these studies, and I think that this is going to give immunologists a lot of really cool stuff to be understanding in the future. So there's one other vaccine type that I want to tell you a little bit about. Yes, I could say a bunch of fun and exciting things about DNA vaccines that I spent a lot of time dealing with, but it's okay. I'm not going to do that right now. It's fine. But the other type of vaccine that I want to mention to you is known as a vector-based vaccine. There are two different vector-based vaccines actually shown here. The idea with a vector-based vaccine is that instead of just taking the nucleic acid, the mRNA, for example, that encodes spike and putting it in a lipid and just giving you the bare nucleic acid, or instead of giving you the bare DNA, you put that nucleic acid inside some other virus. So you use some other harmless virus that does not cause disease to deliver the nucleic acid encoding your antigen. So now you get the benefit of getting this nucleic acid into the cytoplasm of cells so that you're getting PRR stimulation. You're going to get something that's going to amplify the dose, like a live attenuated would. And so you're going to get great immune stimulation and great dose amplification, but you're not actually going to have the risk of a live attenuated of this particular microbe. You can see an example of a vector-based vaccine here. On the right is sort of the general idea, where we can, again, take the gene of interest encoding our antigen, put it into a plasmid, or perhaps put it into some other kind of virus. The Ebola vaccine that has been used to end the Ebola epidemics in West Africa, as well as in a couple of other places, is a viral vector-based vaccine. So this is the Ebola virus. We know that this particular protein is the key antigen against which you need an immune response to. It's called GP. So we just took the part that coded for GP and put it into another virus. This virus is called VSV, or vesicular stomatitis virus. It causes no disease in humans. It causes a little disease in cattle. It's a cattle virus. And so now we have this VSV that has the one Ebola protein on its surface. So you can see the recombinant VSV with the Ebola protein on its surface. We can inject that, and then people will make antibodies against the key Ebola protein. And so the vector-based vaccines have worked really well for Ebola. Also, to be perfectly honest with you, really, if you had asked me in 2019 what was going to be the winner in terms of new vaccine technologies, I actually would not have said DNA. I would have said vectors. There were a lot of vectors that were really close to being over the finish line. And I think that there will still be more vector vaccines coming up. All right. So for the rest of the time, I have kind of some of it's kind of theory stuff to talk about, but sort of important vaccine-related issues to talk about. One of them has to do with the negative side of vaccines. I am as excited about vaccines as anyone you will ever meet. I have been vaccinated against viruses that were eradicated before I was born, aka smallpox, which is the only virus that's been eradicated. But it is important to describe some issues relating to vaccines and kind of what's going on there. And so there are contraindications or reasons why one should not get vaccinated. So we know that there are people who are allergic to certain vaccine components. A number of vaccines, for historical reasons, are made in eggs. And so people who have egg allergy generally don't get vaccinated. If you are severely ill on the day you're supposed to get vaccinated, you generally will wait. If you're immunosuppressed or have HIV infection, we generally won't give you a live vaccine because there is too much risk. And so there are reasons why some people don't get vaccinated. Realize that this is important in our herd immunity calculation. That when we think about, say, 95% of people need to be vaccinated against measles in order to be protected, these people are all included in the 5%. And so it takes a lot of the rest of us to get to that 95%. I'm going to switch the order of the next two slides. The slide that's actually next here is not crazy gory, but is a little gory, so be ready for that. But I'm going to switch the order so it'll be in two slides. And so this is showing some of the data on vaccine complications. And so, yes, there 100% are cases of individuals who have some type of disease or some type of adverse effect after vaccination. 100% absolutely it happens. This table tells us how frequently those types of adverse events happen after someone gets a vaccine, and compares the frequency of similar events after infection. So for example, 1 in a million people who get a measles vaccine get post-infectious encephalomyelitis. 1 in 30,000 people who get a measles vaccine get thrombocytopenia. And that is sad for them. I am not discounting that. However, if you actually get the measles, these are your likelihoods of getting the exact same complications. And so you can see in every case, you are more likely to have a problem if you have measles than if you actually have the measles vaccine. So just be aware, yes, there are absolutely vaccine complications, but this is a risk-benefit analysis. You are taking a risk getting vaccinated of one of these complications, but you're getting the benefit of being protected against the risk of natural disease, which can include, you know, dying. We make that risk-benefit analysis for each of our different vaccines in terms of thinking about what is not sort of acceptable risk. One thing I will also tell you, if you actually look at vaccine trial data, and again, some of this comes from having done vaccine trials. Anything that happens to the patient in the study period gets counted. If you get your vaccine, and then you leave and you get hit by a car, you are counted as a vaccine-induced death. Anything that happens to you medically. This is actually why it's important that you look at both how many people in the vaccine group had the effect and how many in the placebo group had the effect. Some people in the placebo group have a sore arm too after getting a needle there. And so we look as a comparison of these severe events in vaccine groups and in placebo groups and do statistical comparisons. There was one SARS-CoV-2 trial, I think it was a trial in Norway. I don't remember exactly. I think it was this Norway trial. It was a pretty early vaccine trial. And they were like, oh my gosh, look how many people died after getting their SARS-CoV-2 vaccine. If you look at the data, the study population were people in nursing homes who were over 90 years old in Norway. And yes, a bunch of them died after getting their COVID vaccine. And then you looked at how many of the 90-year-olds in nursing homes who didn't get COVID vaccines died, who got placebo. It was the same number. Sometimes 90-year-old people die. And so you have to look at those data very carefully and make comparisons to placebo when you're thinking about this. I will also note that there are some vaccines that we don't currently give. I mentioned that we stopped giving the smallpox vaccine after that virus was eradicated. The smallpox vaccine was largely given, and it was asterisk, largely given the same way that Jenner invented in 1796. One used a forked needle, got a droplet of liquid in between it, poked a hole in your arm and made a circle and squeezed until it bled. And then you had an open infectious sore for 6 weeks, and you have a scar for the rest of your life. You can spread to others. The paperwork that I had to fill out, I was working with similar viruses, so I had to get vaccinated. The paperwork was unbelievable. The whole process is unbelievable, and they told us lots and lots of scary things. So this is sort of how it looks over a month as it tries to heal. I went online to try to find pictures, because honestly, it looked like my arm had been hit by a softball for about 6 weeks. And that's the closest I can see to what I felt like my arm looked like, but it was awful. I hated every minute of it. And if you are immunocompromised, the virus can spread. And they spend a whole lot of time telling us about how you shouldn't touch it and then touch your eye because you can give yourself vaccinia of the eye. And so I spent 6 weeks scared of vaccinia of the eye. And so we don't give this vaccine routinely now. Because once the virus was eradicated, the risk-benefit analysis changed. When this really scary virus was around, this might have been this is necessary to protect you. When the virus isn't there, suddenly this risk is no longer as important. There are currently 2 vaccines that can be given to protect against monkeypox. The World Health Organization renamed it last week. There are 2 vaccines that can be given to protect against mpox. One of them is called ACAM 2000, and it's basically this. There's another one called Jynneos, which is a newer one and does not have this sort of side effect profile. And so whenever I see people talking about monkeypox vaccines and whether we should be giving everyone monkeypox vaccines, and when the health center sent out an email that said Drew was giving monkeypox vaccines to anyone who was available, I sent them a lengthy email asking about which vaccines they were giving. They are giving Jynneos and not ACAM. So again, the risk-benefit analysis is really important. When we think about vaccine complications, there are a couple of other big things that people think about. One of them is related to the inactivated polio vaccine, or IPV. With this vaccine, it's, as I said, an inactivated poliovirus is treated with formaldehyde so that it is not infectious. You then wash the formaldehyde off. So it's not, so some people are like, well, I'm getting dosed with formaldehyde when I get this vaccine. No, we killed the virus with formaldehyde and then we washed off the excess after the virus was dead. There was a clinical trial in 1954 with IPV with 1.8 million children. It had greater than 50% protection. The results were announced in 1955, and it was licensed the same day because people were so excited. And people were so scared of this virus. Lots of kids were paralyzed. The president, FDR, of course, was paralyzed. Here you can see a person in an iron lung. Most polio patients were paralyzed and had trouble with their breathing, the nerves to control breathing, and so they had to have an iron lung to help with breathing. So we can see we were very excited. It was all over the papers. I don't know why this really rude nurse feels like she needs to show it to this patient. Like, that seems so mean. But this vaccine was a huge, revolutionary, big deal. It was amazing. And in about 10 days after licensure, polio started showing up in some of the recipients. So this did not happen during the clinical trial, but then after licensure, there were a number of individuals who started having polio. In the end, there were 260 vaccine-associated cases in both some of these kids and their contacts. All in all, there were 113 people paralyzed and 5 deaths. I am told that there were a lot of pediatricians actually gave themselves the first doses because they didn't want to infect their patients, and that there was sort of this generation of like paralyzed pediatricians, though I obviously do not have the memory to know about that. All of this was because the first lot of the virus done by a company named Cutter Labs was done incorrectly. So when they did their inactivation to make this inactivated virus, they inactivated it wrong. You guys have all done lots of lab work in your life. The thing that went wrong here is that when they filtered, they did a filtration step to get rid of the uninactivated virus, they used the wrong pore size filter. So they just picked the wrong one off the shelf, and as a result, 113 people were paralyzed and 5 people died. So hopefully you understand the importance of paying attention to some of these things in the lab. This situation, the Cutter incident, is the reason why there is something called a vaccine court for people who have vaccine injuries. This is also the reason why there is a vaccine adverse events reporting system, VAERS, where if you have any adverse event with a vaccine, you can go put it into this website. That's sort of a first pass to help collect the data. It is not actually curated at all. It's one of those things where people just put in like, I had a headache after I got my vaccine. And somebody will look and be like, oh wow, 500 people around the United States who didn't know each other had headaches. Like, maybe we should look into that. That's actually how they sort of first wondered about myocarditis and the vaccines. And we're able to say, hey, we're getting some reports of myocarditis. Let's take a pause and actually do some studies on this. Be careful when you look at data on that. As I said, it is not curated in any way. If anyone puts something in there, it stays. A vaccine researcher, tried to make a point of this and wrote that a vaccine turned him into the Incredible Hulk. It took 2 years to get that post removed. So realize there's a little grain of salt you should take those reports with. Of course, there are other issues with vaccines that involve, people making frauds. And that has had some pretty severe issues for the world. But I will stop there, and I will talk about cancer immunology on Wednesday.